Thank you very much. It's really, I want to thank the organizers for this, and I also want to thank all the organizations on this slide. This is my thank you slide. That's why it's been up there for the whole time you've been sitting here. And it's also my disclosure of conflicts of interest, since I owe so much to these companies and government agencies that have allowed us to go from the ivory tower into publicly usable technologies. So this is really about personal genomes and volunteerism in the context of the new World Wide Web opportunities. And I just want to take you through 3 quick slides, putting this in context so we know where we're standing and as introductory comments. This is kind of the timeline of genetics from Gregor Mendel out through the Human Genome Project, and what this is building up to is what happened after 2003. So we have discovery of genetics in the pea plants around the turn of the century, and rediscovery, and then mapping of genes in fruit flies, discovery that DNA is the molecular component of the genetic material. Watson and Crick, of course, discovering the 3-dimensional structure rather than the abstract genetic map, then the genetic code that tells how to get into proteins, and we'll talk about that central dogma from DNA to RNA to proteins. And then Cohen and Boyer with recombinant DNA, which served us very well to get us basically all the way through the Genome Project. Peter just mentioned Sanger and Wally Gilbert developed new sequencing methods, which also carried us all the way through from 77 to around 2005, and is still in common use, but you'll see in a moment how much that's changed. PCR was a key component. Some of the methodologies we'll be talking about. That's a polymerase chain reaction for DNA polymerase. Will make copies of DNA, typically with thermal cycling. And then finally, this series of automation and international efforts at $3 billion cost resulted in sequencing of 1 human genome, actually not even one, no human genome has actually been completed, but success was declared in 2001 and 2002 and so forth every year since then, but we haven't sequenced any yet. So the question is, what happened after this? And what happened after 2003 was really quite remarkable. You're aware of Gordon Moore's law about large-scale integrated circuits, electronics, telecommunications. Computers have all been trucking along at a pretty high exponential rate of a 1.5-fold improvement every year. This is what makes it hard for you to decide when to buy a computer, is every year it gets 1.5 times better. But compare that to what's happened over the last 4 years, where it's gone up, where DNA sequencing has improved by 10-fold every year, and this is due to next generation sequencing or second generation sequencing that kicked in around 2003, 2004. And it kind of harks back to some of the work that we did on multiplex sequencing in the 80s, but then Rob Mitra's electrical engineering graduate student joined and took this idea of basically taking an ordinary glass microscope slide and putting single molecules of DNA scattered on it dilutely and turning each of those molecules into a colony, like a bacterial colony, except the colony is formed by polymerase, an entirely in vitro reaction where this enzyme makes copies, and each dot that was a single molecule turns into a dot which you can sequence. And you can sequence it in at least 2 different ways. These dots can be sequenced either with polymerase, the same sort of polymerase is used to make the colony, or ligase, another enzyme, which both of these have in common that they're exquisitely sensitive to mismatches, either with single nucleotide mismatches here, where you have 4 different fluorescently colored nucleotides, A, C, G, or T, with 4 different colors, and the enzyme polymerase will only incorporate the correct next one at that position. Or ligase will take in now the same kind of color coding, but now with a long piece of, or a nine nucleotide long piece, where one position is coded G, C, A, or T with the colors. So these two different methods are in use in various commercial platforms like Applied Biosystems, Illumina, and so on. And there are about 14 companies now that are in this field, and it's actually, the price has dropped largely because it's now not a monopoly, but it's a field full of exciting ideas and new instruments and a lot of sharing of ideas. And some of these, I don't want to go through them all, but they have different read lengths, and they have, some of the costs are as low as $5,000 for human genome, down from $3 billion. And so there's quite a bit of progress here. And I just want to focus on one little aspect of this, which is the idea of open source. And that open source is part of the two-part theme of the title of this talk, which is how can we utilize the sharing that occurs in Web 2.0, and it often occurs in science. And we'll get to this illustrating here. Rich Terry is the head engineer on this, where the research and development was done at Harvard, but it's done in collaboration with Danaher, a company that makes motion control for semiconductor industry. But the entire object, the entire device is described in detail, and users are encouraged, the experts are encouraged to take it apart and put on new parts. It's really designed to be reconfigured. And the hardware, software, and wetware are intended to be open architecture, kind of like the IBM PC or some of the Google tools, where they encourage a community participation without penalty. And the price of this is about 4 times lower than most of the other instruments, again, to encourage people to not be intimidated by it. Now, if we have all these great systems, why do we need an open architecture? Why do we need to keep developing? And the reason is that we keep thinking of new things, not just new ways of doing DNA sequencing, but new things that you can do by integrating DNA sequencing with other things you do in the laboratory. And in this example, we can add on to this device a very simple device, just the digital micromirror that might be in that projector that's projecting there, where each of these, you have a megapixel of little mirrors, each under transistor control, which reflects light onto the flow cell. We can either have a flow cell of chemical synthesis or a flow cell of enzymatic or cellular reactions. And this allows us to change the sequencer into something that can do fabrication, synthetic biology. It can do sorting of cells. It can do sorting of DNA molecules on beads. It can do a whole variety of things just by adding to a $170,000 device a $5,000 digital micromirror. So that's just an example, and we want to encourage people to think of other examples and be unintimidated by the device itself. And this will be the theme of the open source, is to be not only unintimidated by the hardware, software, and wetware of the sequencing, but also of the information that we're getting from personal genomics. And to illustrate this idea of being unintimidated, demystifying, destigmatizing, and you can ask one of the hardest questions, which is, what if you discover something about yourself or your family for which there's no cure? Wouldn't we be better off not knowing that? And that's a perfectly valid response for many people, but there are many people who take a different tack, and these are various groups and individuals who've decided that they want to talk about it openly. It's non-anonymous. They actually say, our family has this problem, and what they're doing is becoming activists, biological activists. So we have, for example, here's Nick Nolte and Susan Sarandon in a kind of a typical Hollywood blockbuster on a typical subject of lipid biochemistry. And they didn't know much lipid biochemistry when they started out, but their kid had this inherited disease which was new, and they learned enough about it, and they raised community consciousness and awareness and raised money and actually contributed personally to the nutraceutical that definitely helps in this case. And this is one of my colleagues, a Harvard professor whose family has diabetes, and so that prompted him to launch a difficult and unprecedented effort to get human stem cells working in the lab despite political opposition. And Michael J. Fox has very publicly come out for Parkinson's disease, and Hugh Reinhoff is here on the cover of Nature with his daughter, and he's trying to figure out what she has. It seems to be a new syndrome. And the Haywood family, one of the brothers here had ALS, and they established patients like me to allow people who had not just ALS, but a variety of neurodegenerative diseases, psychiatric diseases, AIDS, things that you would think would be very stigmatizing, the things that you might want to hide from the world, but they decided that the benefits to the community of other people that had this would outweigh the risks to themselves, and so they share very freely their symptoms, their name, their face, what treatments they're taking, what drugs, and so forth. Nancy Wexler, Betty Ford, you get the idea, is that there is action that you can take in every case. On a lighter note, you can take action by buying your kids a do-it-yourself biology kit, DNA Explorer, for $80, and they can use this to, take some saliva and sequence their DNA and find out who their father really is, and other innocent activities for your kids. Or the National Geographic, in collaboration with IBM, has allowed you to trace your roots all the way back to Africa through this circuitous route. And 23andMe got the invention of the year for this tube that you can spit into. So that's a great invention. And home tests range all these things you can do, steroid testing and stress and health hazards and pregnancy tests, and all these things are moving some of the medicine out of the clinic and into your home. And of course, each of these things needs to be carefully regulated, but it does indicate something to pay attention to. And Google and Microsoft are launching personally controlled health records, another example of something that normally would be something you would say would be clinical. They're helping you gain control so it becomes more portable and you have access to your own data. So thinking in this sort of theme of home testing and being advocates has led people to critique this, and spurred on by a New England Journal of Medicine, a set of 4 articles, Nicholas Wade writing for the New York Times, a fairly balanced text, but with a fairly inflammatory title, which is Genes Show Limited Value in Predicting Diseases, which kind of erases decades of work where clinical geneticists still get high value, and you'll see in the next slide. And so I included here the actual text from Mark Twain saying the report of my death was an exaggeration. And the report of the death of genetics was a bit of an exaggeration too, because there are this is a rapidly increasing number, but 1,449 diseases for which are adequately predictive and medically actionable enough that clinical geneticists will order these up. Now, these are not the direct-to-consumer tests that I showed in the previous slides. These are things where they will PCR up, amplify up a little piece of one of your genes for $2,000 and sequence it, not do SNP analysis, single nucleotide polymorphism analysis, but they actually do some kind of very thorough sequence or functional test for diseases such as phenylketonuria, hemochromatosis, Tay-Sachs, cystic fibrosis, and so forth. Now, phenylketonuria is an example where this is 100% heritable in that if you have it, you don't do anything about it if you eat normal food, you'll be severely mentally retarded. On the other hand, it's 100% environmental in that if you do eliminate phenylalanine from your child's diet, then the child will be absolutely normal intelligence. And so 4 million, almost all children born in hospitals in the United States, 4 million per year, are tested for this. And it's relatively, it's common enough that it's important to catch it. And Tay-Sachs and some of these breast cancer genes are highly enriched in certain populations in the world, such as the Ashkenazi Jewish descendants. And so they are very careful to look for this premaritally, actually, before they make decisions that could result in very painful children in great pain. And even drug companies, although they don't really want their markets to be fractionated into little pieces, occasionally and increasingly their drugs are more efficacious, they work better, they're less toxic if they can target them for a particular set of people who will benefit from them and will not have a toxic reaction. And examples of this are Herceptin and Iressa. And these, you basically have a drug and a diagnostic that work together. And even though most of the slide is talking about medical consequences of personal genomes, there's also ancestry, forensics, identification of individuals from small amounts of DNA, social networking, education, and research. Some of these are reflected in companies like 23andMe, in addition to the medical or modifying the way we look at medical. Now, the theme that I'm developing here is the idea of open architecture, of shared information, and some wonderful examples we have. I already mentioned the IBM PC, the World Wide Web, many people use that. Linux operating system as an alternative to Microsoft monopoly, and Wikipedia. And then there are examples of closed architectures which also have their place in society, but we need to explore both of them, and I'm going to mainly be exploring the open architecture. And we had for the Genome Project, we had a huge effort to get the data out as quickly as possible. Within 1 week of getting the data, we would put it onto the internet so that people could look at it before we even had a chance to publish it and think about it and patent it and so forth. And that was a conscious decision. But as we start moving from a generic human genome into where it really matters is when we start connecting individual genomes with the individual traits. And it's no longer generic, it's personal genomics. We now have these two important ideas, privacy on the one side and sharing on the other, and they come into apparent conflict. And I say apparent from reasons that you'll see in a moment, because there might be alternatives. But for emphasizing the openness, we actually have laws requiring all NIH-funded research be open access publishing. So that's clearly a trend that's increasing as of just December 2007. And then on the other side, attempts to put information in the public domain, de-identified, by, say, pooling lots of individuals, putting the cases in one pool and the controls in the other, no identifying features. Some computer scientists showed that this was sufficient to identify who was in the cases and who was in controls, which is not what they wanted, and so they pulled all this data back out of the public domain, and that's mildly embarrassing, and it also makes it less useful. And computer scientists identified the governor of Massachusetts and his family's health records from open access information. And the point is that there are a large number of ways where either your DNA or your trait data are identifying, and you need to be clear on this when you're communicating it to the research subjects that might be interested. Here's some other data, like iris scanning might be considered a phenotype, a medical phenotype, but it's also used for identification. Fingerprints, dermatoglyphs are actually used in some diseases. And DNA, of course, is used in there's 13 CODIS sites in the DNA that used in the United States for identifying individuals based on just trace amounts of DNA left at crime scenes. So this list has 12 items on it. They're not hypothetical, they're actually real, and one of my favorites is number 5 here, where a 15-year-old kid who was the product of an anonymous sperm donor. His mother knew a trace, very tiny amount on a need-to-know basis, but really didn't. It was anonymous, and the son decided that he wanted to know who his father was, and he took a cheek swab and sent it into an ancestry service where the Y chromosome and the surname, their last name, track going back in history with the paternal lineages, and he was able to find his surname through this service and identified his father correctly on the first try, and basically showed up on his doorstep unannounced saying, "Hi, Dad." And so obviously not everybody is as talented as 15-year-olds, but it's still quite likely that a variety of people could do this. And we won't go through the whole list, but there's things, abandoned DNA and government subpoenas are ways that these things can make it into the jurisprudence. All of this boiled down to, we felt that there was a need for looking at other ways of consenting individuals so that we would specifically get people who understood all these risks and were okay with it. And so the Personal Genome Project is the first and unfortunately the only open access database right now that combines human genes, environments, and trait data, and makes the cell lines for each of these individuals available to researchers all over the world with no strings attached. So in order to do this, and based on all the things that I've been showing in previous slides, we want to avoid over-promising on de-identification, making it anonymous, because we felt that that was not something that we could necessarily, it's not a promise that we could keep. And so we make that very explicit. And in order to make sure that people understand this, we have an exam, an entrance exam, and you have to get 100% on it, which is not very hard. We think it's high school education is adequate. And you get to take it as many times as you want. But the idea is to educate people involved in medical research before they get involved, rather than after we discover something about them and have to tell them, and then they tell us, oh, we didn't want to know that. Well, it would be better to have found that out at the beginning and just keep them from participating. So just a show of hands, how many people here have actually participated in medical research before? It's been a research subject. So I have, okay. That's actually a very high frequency. I think that's quite nice. And so, but clearly, it's not for everyone, medical research in general in this project. So if you're interested, go to personalgenomes.org. We're trying to also get a cohort, which is a large cohort, where we're getting multiple traits, not just focusing in on this study is about osteoporosis, and we're only going to study the osteoporosis candidate genes. It's really a broad set of genes, the whole genome, and a broad set of traits, because one person in the cohort will be a case, meaning they'll have a trait, another one will be a control, or they'll have a different quantitative trait, and they'll fall a different point on the bell curve. So they're all interesting for something. These cell lines are available. The numbers of the cell lines are underneath each of the faces here, and we have IRB approval to expand this to 100,000 volunteers. And we have about 13,000 right now working their way through the queue, working on the exam. Now, the point of this is to collect data from the individuals, not to give information back to them. But unlike many previous studies, we decided not to, there's an increasing trend to acknowledge that if you find out something that's highly predictive, highly actionable, and life-threatening, then you should need to give that information back. And it used to be that they would say, even under those circumstances, we won't give you your information, we won't tell you about what we discovered. So we're trying to hit a balance between, we don't want to be a service and we don't want to promise that we're doing clinical research, a clinical service, we're just doing research. But just in case, we do want to look for alleles that might be actionable. And here's a case which is kind of in a gray zone. We've done in order to make this possible for us to fairly automatically look for alarming sequences in these genomes, we have about 17 full and partial genomes that we've done. And one of them turned up, as an example here, hypertrophic cardiomyopathy. And this is fancy medical jargon for the wall of your ventricle of your heart enlarges, and if you're exercising without any warning, you can drop dead during exercise. Even athletes drop dead on the basketball court from this. So it's something that you wouldn't dream to ask your physician normally, but if you have it, it's good to actually find out whether your heart wall has enlarged. Now there are many different ways you can get this disease, many different genes, many different ways of changing each gene. This particular one that the PGP number 6 had, it's the mutation was an A at position 13 to a T. That mutation was only found once in the literature. We searched through all the databases and all the literature. We found one example, and that individual had hypertrophic cardiomyopathy, and clearly all the normal people that didn't have it. So you could consider this a case and all the others a control, but it really wasn't a design study, and the number is too small to have any kind of confidence that this is an association. But we felt, and so a number of 1 is not really enough to worry this individual about it, but we felt like we should follow up a little bit, and it turns out that the clinical geneticists on the staff of the PGP know that if you ask, then ask around, then you will find out there's a lot more information than is available in the literature and in databases. So they ask around to their friends, other clinical geneticists and genetic counselors, and found a very large number of cases here, these black squares and circles, which represent additional cases of this same previously very rare allele, and now many cases of having the allele and having the disease. We felt that this was not airtight, it was not entirely predictive, but it was enough evidence that accumulated that we should ask this person or recommend to this person that he talk to a physician about it. And so he went to a cardiologist, got an echocardiogram and an EKG, and the good news is he's so far symptom-free, but the cardiologist feels that he should have a checkup every 2 years to make sure that the ventricle wall doesn't get larger. So this is a kind of an example from the cutting edge of personal genomics, where you get something which is fairly new, and you have to expand it a little bit in order to justify telling it to somebody. In many cases, there will be something which isn't justified, and the patients have access to their full genome, but you may or may not want to alarm them about it. And it illustrates what we might call a systems biology challenge, the human body and genetics being a large, complicated system. And we would love to be able to go from this complicated genome with 6 billion base pairs to the complicated traits, but we don't feel that we can right now or that we have to. So we can do quite a bit more than merely predict. We can actually measure quite a bit and bridge this gap and make our predictions on much firmer ground. And the way we do this is we don't just look at the inherited genome from your mother and your father, which you might do once in a lifetime, but we collect information on how the genome plays out with the environment here, your microorganisms that are in your environment, allergens, viruses, fungi, bacteria, and so forth, and your immune response to them, which we'll call the VDJome for technical reasons, but immune response to the microorganisms, both of these reflect your environment. And then, in addition, how your genome plays out, you have protein coding regions that change the function of your genes, and then another set of regulatory mutations that affect the level. So you can have the level of the gene products, the proteins, and changing their functionality. So if you bridge these gaps here, then you get better, closer to what we might expect personalized medicine to do, where when you go into the clinic, you know exactly already maybe what microorganisms you have, what drugs they're resistant to, and how your personal genome is playing out on a day-to-day basis. Now, as an example of that measurement of the environment, which is heavily dependent upon the particular genome that you have, and the particular environment you have. It's a perfect example of this interplay of genome and environment. Morton Sommer and Gautam Dantas in the lab did a survey of 18 different antibiotics. So in a way, we could just sequence every microorganism, every virus, every allergen in your body. That would be an even bigger task than sequencing your genome. And it wouldn't necessarily tell you that a tiny fraction of that would be clinically relevant. But one thing that we know is clinically relevant is whether your microorganisms are resistant to particular drugs, because there's no reason to expose you to drugs that aren't going to work for you and might be toxic. And so we call this kind of enterprise a bioweather map because we think that average citizens can be excited about weather. They understand weather. It's a type of science that connects them. It's immediately useful and understandable. And we feel the same thing should be true of, as you see a cold front moving in with high or low pressure, you also see a cold front moving in with rhinovirus or coronavirus. And this should be something that people can follow, the consequences of sending their kid to daycare and so forth. Anyway, back to the 18 antibiotics. This is almost most of the major classes that are used. And we sampled environmentally from human oral and gut microbes and from environments which were far away from cities and from farms where there shouldn't be any exposure to these microorganisms. And this is a phylogenetic tree, which is basically, you can think of kind of a family tree of microorganisms, where the further you are from one other the further you have to go down one of these lines and back up, means the more distantly related they are in this family tree that goes, basically it's a tree of all living species. And what's interesting is these are color-coded with all the different antibiotics, and almost every antibiotic has representatives of multiple different distantly related families of bacteria, and almost every one of these bacteria are resistant to one or more antibiotics. In fact, a very common outcome in this particular study is if they would be resistant to almost all of the antibiotics. And not only were they resistant to almost all the antibiotics, they would actually eat the antibiotics. They would actually subsist on the antibiotics as their sole source of food. And I could hear from your response that you were as surprised and amused about this and scared as we are. And so we're following up on this in a variety of ways. Some of these might be new ways of being antibiotic resistance, or they might just help us understand the old ways. But it's very important these are living on antibiotics at almost 100 times the normal medical dose that is enough to kill pathogens. Now that's what the microorganisms you see in your body can spike. You'll get a microorganism coming in your body, and it'll be there for a very short period of time as it gets cleared out by dilution or by immune response or something like that, and it's gone. And so you can't blink. If you're doing a molecular assay, you have to get the timing just right or take a lot of time points. On the other hand, if you follow your immune response to it, the immune response comes up and then stays up for years, sometimes decades, and so you have a greater opportunity to see that you've been exposed to something. So this is the basis of the HIV test, for example. If you've been exposed to HIV or tuberculosis, you can detect this by your immune response to it. And your immune response, or at least part of it, is composed of this kind of textbook Y-shaped molecule which has 2 light and 2 heavy chains, and each of these little black bars is a portion of the chain that's just hypervariable, changes quite radically because there are multiple copies of the genes, each with its own type of reactivity, and these are combined. It's one of the few genes in your whole genome that actually changes from cell to cell, and it changes very dynamically and is selected for responsiveness to microorganisms or vaccines that you're being exposed to. And there are millions of combinations that can occur. And so we would like to be able to do is sequence these hypervariable regions, these black bars here, and that might change from day to day, week to week. And as an example, here's a vaccine experiment that we're running where we're tracking the human dynamic response to 11 strains of viruses and bacteria, such as last year's H1N1 flu vaccine, hepatitis, polio, yellow fever, and so forth. And what happens with each of these are adult vaccines because they're adult volunteers, not the childhood ones. And what happens is for almost any antigen, you have a low level of antibodies already present. You've never seen this antigen before, but they're there because you've made all these combinations in your blood or in your bone marrow. And they're very low levels. And then you give it, you either get exposed to the pathogen or you get a vaccine, and then in a few days, you get a primary response, which is about 1,000 times higher. And then if you get a booster or get exposed to the organism again later, you get a response which is another 1,000 times, so 1,000,000 times higher. And so you should be able to detect this amidst the noise of all the other things that are going on. And we think this, again, is an example of personal genomics, which goes beyond what you inherited from your mother and your father. So that's the environment, but what about what we call the epigenome, which is maybe a little hard to predict from your genome, but it's important. It's something, your genome is present in every one of your cells, fairly much the same except for your immune system. And even there, pretty much the same. But your epigenome is how it plays out. In other words, how it's regulated and how the levels of all your RNAs going from DNA to protein change. And this is regulated by methylation and protein binding and so forth. And this helps us bridge the gap between your personal genome and your traits via your environment. And just a quick textbook refresher is what we're calling the epigenome is this whole transit, or sometimes called the central dogma, where you go from DNA to RNA to protein. You get DNA that can replicate, being transcribed by this little yellow polymerase, just like the polymerase I mentioned earlier, to make RNA that goes through various processing steps, leaves the nucleus, this blue nucleus, goes out and binds to these ribosomes, makes protein, the little green proteins. And there's other RNAs that are made that are needed for translation, needed for regulation. The proteins themselves can be regulatory. So this whole regulatory network is sometimes called the epigenome. Now, to get at the epigenome, you might want to collect a very large number of samples. It's not just your one genome, it's different time points, different tissues, different cell types, and so forth. And so we do this barcoding. It's just like barcodes in the stores, except now it's molecular barcoding. You're barcoding with DNA that allows you to that every time you take a new sample, you immediately barcode it so you don't lose track of it. And no matter where it goes, no matter how you sequence it, you know that came from that person, that cell type, that day. And there are many different kinds of RNAs. There are RNAs that code for protein. There's regulatory RNAs, like so-called microRNAs. And we've developed ways to efficiently barcode via oligonucleotide adenylation, just jargon for preventing them from forming these circles, which is what you don't want, and getting this barcode in red attached to your whole collection of microRNAs in yellow. RNAs don't just go straight from your genome to translate into proteins. Sometimes they get edited. These aren't really mutations, these are programmed editing. There were a very small number of these that were known when Erez Levinon started looking at this both computationally and experimentally, and he hypothesized there would be a lot more, and he would try to figure out the rules from the first 10, and it allowed him to discover 569 of them, and that number will probably go up in the future, where you see a mixture of the original A and a G, which is introduced, edited into the RNA on its way to proteins. Another kind of regulatory phenomenon, just kind of give you a feeling from, this is from the cutting edge of science and personal genomics, is you can look at Madeline Ball looked at the methylation, which is related to expression level, by this trend, this rule that high expression of the RNAs corresponds to is known to correspond to low promoter methylation, but also to high gene body methylation. So you can see this really dramatic dip for the red RNA species, which are high expression from the high levels of expression, relative to the blue, which is low levels. And so you can see these kind of trends when you develop genome-wide assays. And again, these vary from day to day and tissue to tissue. We do selective sequencing to do this, where we can take a little red piece of synthetic DNA to fish out the yellow parts of the genome or the transcriptome that you're particularly interested in, the RNA. And by either gap fill-in ligation or hybridization selection, we can pull out literally hundreds of thousands of different parts of the genome. You can either sequence the whole thing or go in and pick selected parts, like say the drug resistance components of all the microorganisms. So you don't have to sequence everything. But you can put in 258,000 different, think of them as molecular tweezers in one tube of solution. And this is Kun Zhang, who's now at UCSD, did a lot of this work and figured out how to do this and refine the methods without going broke buying 258,000 oligos every time he changed his protocol. And the reason that this was inexpensive, you can kind of get the theme here, which we make oligonucleotides on arrays, and then we can strip those oligonucleotides off the arrays, so we can make hundreds of thousands in a automated manner. And we don't need to go through all the details, but you can basically amplify them because they're in trace amounts, and then chew off the ends of the amplification, and then these things will circularize, and you could fill in a ligate. And here are these barcodes again. You can introduce DNA barcodes on the fly to identify the patients and the samples. So the good news about this very simple plot really is as you increase the number of genes that you're considering for an association, a correlation between your genome and how it plays out, causing your various traits, your genetic inheritance and diseases and so forth. As you said, there are 3 million differences between you and the reference sequence. And those 3 million differences, any one of them could be causing your disease or your diseases or your traits that might not be diseases. And so each of those is a separate hypothesis, an idea that you want to test. And the more ideas you want to test, the weaker your statistics become. But the good news is even as the number of ideas you want to test, all sorts of combinations of alleles, genes, environment combinations, even as that goes up into the Avogadro's number of hypotheses, of ideas, 10 to the 22nd, billions and trillions of ideas, the number of genes goes up, the number of, individuals you need to recruit in order to get statistical power goes up very slowly, logarithmically, and this is good. You still need a lot of people, 100, here thousands, but for lots of rare diseases and low risk ratios, you need lots. And it might be the number of hypotheses that we had in the previous slide, I said it might be 3 million, or 1 million, or 300 per gene, or maybe you can lump them in various ways with gene sets. But it's a large number of hypotheses. And when you finally get a correlation between genes and traits, you might want to prove it. You want to move from association or correlation to causation. And just give you some flavor, this is not the only way to do it, there are many ways. But one of the ways is you can engineer mammalian genomes. And mammalian could mean you can engineer your own human personal stem cells, we'll get to in a moment, or you can engineer a closely related species to see if you can reproduce the trait itself or some surrogate for the trait, some cell-based or organism-based trait. And so here's an example where very interesting trait, little off topic, but the mouse lives for about 2.5 years, kind of illustrates how how really age is a planned obsolescence, because a very closely related rodent called the naked mole rat will live for 28 years. In addition, it has skin that is pain tolerant. It can tolerate high acid buildup that happens because it's very carbon dioxide tolerant, because it lives in close quarters underground. And it lives more than 10 times longer than its close relative. And so by engineering their genomes, you might be able to figure out what, over a long period of time, it's hard work to figure out what genes in the naked mole rat can be introduced into the mouse to make it live longer, or any of the other traits. Now, to get at, I don't want to belabor this point, but to get at the differences between mutations that cause a change in the protein and mutations that cause the level of the protein to go up or the level of the RNAs to go up, you look, they're called these Cis Elements. These are regulatory elements that act only on the strand of DNA to which they're attached, and usually in close proximity. So if you think of these two strands of DNA, one was that you inherited from your father on the top, and one you got from your mother on the bottom. And there's a trick that you can use where you just look at any arbitrary point that differs between your mother and your father's copy in each of your cells. By monitoring that and monitoring the ratio, the number of counts of each molecule that you obtain, because they're going to be expressed at different levels, if there's anything in cis that changes that level, you'll find that out, and you can use this to either discover the cause and effect relationship, even with a very small number of examples. I kind of exaggerating this by saying n equals 1. You can go and you can test the hypothesis we did in the previous slide. Now, if you're going to, the thing about epigenomics is genomics, reading DNA sequence, you can do from any cell. Doing the RNA or the epigenomics, you have to do from every cell. And so, can you get every cell from a drop of blood? Not really. If you're looking for one cell, we can sequence from one cell. We can even get close to whole genome from a single cell. But there's a big difference between there's some enthusiasm for using scaling down technology so that this is enough. You can certainly do a glucose test from this, but doing genomics on rare cells, and I'll show you in the next slide what I mean by rare cells, 6 microliters that you might get from a drop is very different from the 6 liters of blood that you have floating through your whole body. And what are rare cells? You have circulating tumor cells that come and go. Some of them never amount to much of anything. Others are forming metastases. You have pathogens. You have fetal cells. If you've been pregnant, you will often have fetal cells from your baby for many years afterwards. And these can be separated out from the blood. In fact, we're moving to a time, we're not quite there yet, where you could actually pass your entire 6 liters of blood through a device and get 80% extraction. And this is there's already a method here which is 1,000 times faster and 80% extraction of these sorts of cells using a magnetic procedure. This is just an idea of where we might be going in terms of being able to monitoring and in some cases clearing your body of these rare cells. But another way of accessing your cells is by taking one of your cells and reprogramming it. This is a different one. Now, we're not looking at the real cells that are in your body in their normal state. We're taking one cell, in this case, or one sample, rather than doing a biopsy of every cell in your body, leaving you with no cells, which would be not be good for our volunteerism. Instead, we take, say, pluck a hair, and that has enough cells in it to establish an embryo-like state, reprogram the hair so it's more like an embryo, and then you can generate from that. We and many other labs can generate all the major developmental phases, ectoderm, mesoderm, endoderm, imperfectly right now, but increasingly there's a big community developing this, including In-Hun Park and George Daly, our collaborators, and Jay Lee in my lab, that can be used either for developing cell therapies, or in this case, in the Personal Genome Project, developing diagnostics and personalized medicine. And I just want to conclude on some example of some data that you get from this. I know this is a very complicated slide. These little trees are like the phylogenetic tree I showed for bacteria, but here we're classifying the response, the expression level, how your DNA plays out in terms of RNA and protein. For a variety of individuals, these PGP1 and 9, for example, different biopsy sites, different reprogramming them from skin cells to fibroblasts to induced pluripotent stem cells, which have a much broader capability than skin cells, and then to embryo-like differentiated tissues. And the take-home for all this is that when you look at expression levels alone, in other words, the absolute amount of RNA that's made for each of these different genes, there's a tendency for the expression to correlate with the different tissues. So the blood cells from PGP1 and PGP9 look like each other more than the skin cells from PGP1 and PGP9. But if you look at allele-specific expression, at least half of them are invariant between different cell types. So you get the same, the ratio of your mother and your father's expression, how they influence your expression, it segregates or associates with the person. So now they cluster by person rather than by tissue type. And this is sort of what you would expect, but it's quite interesting. And that means for 50% of these, you can use almost any cell. And for 50% of them, you have to look at a variety of different cells to get the allele-specific expression. So you can think of it as two kinds of expression. The absolute amount of RNA that's made, and the other is the ratio of the amount of RNA that's made from your mother and your father's copy in your cells. And so that's a very complicated ending to what is almost certainly a big change in the way we're looking at genomes, from going the generic genome to personal genomes. And just to summarize and start you thinking about, this forum is to have a discussion, to have question and answers. This is where we've been in this talk, where we've got this personal genome where there's 3 million differences between you and the reference sequence, or you and anyone else in the room. And we don't try to go straight to traits, but go through this epigenomic middle that modulates, regulates the level of the RNA and protein, and keeping in mind that this doesn't happen in a vacuum, it happens in an environment. And just looking at allergens, microbes, and viruses is not your entire environment. There's also the chemical environment, your educational, your cultural environment. And we try to capture as much of this as possible through questionnaires in the Personal Genome Project. So period, full stop, and I hope I've stimulated you to ask questions, troubling or otherwise. Thank you very much.
Thank you, George, for a fascinating talk. For the question, I please ask you to take the microphones. We have 3 microphones in the audience. I also ask you kindly to fill out the surveys. It's important that we get feedback, and I'm sure they're interesting questions. Who wants to start? Back in...
Just one back there.
Hi. I was wondering, do you think there will ever be a personal epigenome project?
Yeah, so the question of a personal, in a way, this is a personal epigenome project. We are collecting from each individual some cells. So, for example, skin or hair cells, from which we can reprogram their cells and determine how the epigenetics plays out. And to some extent, we could get cells. We get cells from blood and saliva and so forth. If you have in mind getting epigenetics from all their naturally occurring cells, which includes the environmental insults or benefits that they provided to the actual cells in your body, that's a much more difficult problem, and probably should be coupled with some protocol that includes end-of-life analyses. And I think we're not there yet. I think we're more interested in a prospective study, starting with people that are not at the end of their life. Question over here.
How long has it taken you to get the... Oh, sure.
How long have you been organizing the project? You say you have 13,000 participants or people in the process. How long do you think it will take to get to 100,000? And how important is it to get to 100,000?
So the question is, how long has it taken? How long will it take? So we're trying not to say, so we started the very slow process of getting approval and getting all of the infrastructure in place that's needed both to do it technically and to do it in a way that's responsible socially. We started that sort of process in around 2004, 2005, and really only this spring did we get the full permission to expand to 100,000. We kind of opened the doors on April 25th, which is DNA Day. Now we already had 10 as a kind of a worked example on the internet so they could see what it sort of looked like with 10 real people. We don't know exactly how long it will take the process. We're doing it in factors of 10, so the next set is 100. As soon as we're done with 100, we'll go to 1,000 and 10,000. And we think it's scalable, and we think it will go quite quickly. We have constantly getting donations from companies donating their time and their goods and services. And it's really wonderful to see this level of altruism. But I don't know. As an example of how quickly it could go if we were doing something simpler is the Genographic Project. In about 8 months, they went from 0 to 100,000. That's a much simpler process. They were just looking at 12 different places in the genome. We're looking at all 6 billion places in the genome. So, but there's been a lot of technological progress since then as well.
Have there been any recent changes and improvements in the computer technology that allows you to store and retrieve all this information that you're gathering?
Right. So these new generation of sequencing instruments produce a truly prodigious amount of information on the order of terabytes of information, 10 to the 12th bytes of information per day. Most of that in image data that we can throw away. Some people store it, but it creates a mess. Definitely the disk drive technology has improved very significantly. And this whole project would have been very difficult to do, say, a decade ago. The DNA sequencing is going much faster than the computer improvement, as I already mentioned, but it's still quite manageable with current computing if you set things up properly. But we have Isilon is a disk drive manufacturing management system that has donated their time and effort. Amazon.com has offered to host the Personal Genome Project for free in its cloud computing infrastructure. Google has helped us. There's just a lot of volunteerism going on at that level as well. Yeah.
There's a question here in the middle.
And there's one here.
How do you expect the personal genome interplace with the social networking, say in the next 3 to 5 years? What do you think will happen?
I'm sorry, you'll have to repeat that. I can't hear it.
How do you expect the personal genome interplace with the social networking? In the next 3 to 5 years, what would you expect to happen? Do you think privacy would be a major concern?
So I think the question is how the Personal Genome Project interact with social networking and playing out in the next few years. Well, we think it's an important component. You could think of it like Wikipedia, where in principle, no one has to prove they're an expert to contribute to Wikipedia. There's a crowdsourcing and cross-checking and policing that goes on that doesn't depend on degrees and so forth, but successively approximates a really quite high-quality encyclopedia. And here is even more of a case where you are the expert about your traits more than any scientist is, and you need to share that. It's not something a scientist can go off and do by themselves with their grant and their rodents. They really have to interact with the public, and the public has to be willing, at least some small subset of the public has to be willing to share with each other and with the scientists. So I think you can think of this as a kind of social networking. There will also be a component of social networking which will spread the word. We want to nucleate it from a very diverse set, geographically, socioeconomically, ethnically diverse, and different disease communities, to nucleate it and then spread by word of mouth. If they found the project intellectually stimulating or personally helpful in some way, then they can recruit additional people. I think it's a very important component of this, much more important than usual science, where you can kind of just work by yourself in your laboratory. This is very much a public phenomenon. Question over here.
Hi, thanks. Can you comment on how making individual genetic information public or open source might actually stimulate us as a society to develop the instruments needed to protect people against the misuse of that information?
Right. So having an open dialogue about the risks and the benefits of being able to share for research purposes, and to some extent it is valuable, and to help people visualize that a lot of people have this. Some people will have the allele and no symptoms, and other people will have the allele and have symptoms, and the ones with no symptoms, they might be doing something interesting in terms of their lifestyle that doesn't require development of new drugs, maybe just nutritional or something like that. And whenever you find that, however rare that is, that's incredibly valuable immediately, in that you can try it out on the ones that might have symptoms of their family. From that context, insurance companies and even employers might find that it's in their best interest and in their community's best interest to provide, either free or for nominal costs, access for everybody, all employees are insured, their own genome, so that the employer and the insurer doesn't see it. They provide it, or they provide money for it, but they don't see it. They help you get access to software, they'll help you interpret it, access to your physicians get access to it. And then you can start using the accumulating information that's making a more personalized medicine. It should reduce the healthcare costs, and that should be a win-win situation for both the individual and the insurance company. But it requires, if we each hang on to our data, we all lose, basically. Not everybody has to share, but somebody has to share. Yeah. We have hold up your hand if you have a microphone. There you go.
I have a question. So with more people being able to be involved in their own genomes and their sequencing, what do you think about people patenting genes and genome mutations?
I think I'm going to ask you to repeat that. It had to do with patenting the gene and looking at your own genome.
Well, because people are becoming, as they become more involved, they're actually like owning and involved in their own genome sequencing. Yeah. What do you think about people and companies patenting certain genes? And how
Okay.
that
Right. Okay. So there is kind of a very interesting question that you're raising about if a company like Myriad Genetics is the one that often comes up. They patented 2 very important breast cancer genes. They deserve a lot of credit for that. They even deserve a lot of money for that. But it could be perceived as a barrier to progress, in particular if in a research tool like this, we're getting access to their gene and lots of other genes. We can't afford to be paying each one of them, $2,000, and it would be millions of dollars per individual. Whether you get a positive result or a negative result, in principle, they charge you $2,000 in either case. So that's clearly a non-starter for research, and I think it's not their intention to interfere with that kind of work, and it's not our intention to interfere with their business model. In fact, we genuinely hope that if there is a positive in heaven forbid, in any of the individuals in our project for one of the 2 breast cancer genes that they use, that they will confirm that by taking a clinical version of this. In a way, they've been alerted, but they really should get the test from Myriad. Moreover, all of the people in the family should consider doing that. So rather than stealing business away from these patent holders, we may actually be generating business for them. So that's one way of looking at it, is they shouldn't be threatened by our project. We shouldn't be worrying about paying $2,000 for each of 1,400 genes. But the other way of looking at it is probably there will be a trend in society to reduce those costs and figure out other ways for these companies to make money than patenting every single interesting allele that's in the genome. It really should be getting close at this point to free. It should really be part of the healthcare system. And I'm sure there will be all sorts of creative business models for allowing us all to innovate and invent without burdening the medical system. There's a microphone here, and you have a microphone? Yeah. Go ahead and start.
In the way of prevention, what sort of things should we be anticipating?
Prevention, did you say? The preventative part of personalized medicine, is that the question? Yeah. So, well, phenylketonuria is a classic example where if a baby has this, then your prevention is you get them immediately on a very extreme diet without any phenylalanine. So they can't drink, diet soft drinks that have NutraSweet, any phenylalanine derivatives. They can't eat proteins that have phenylalanine. Other preventatives, if you have hypertrophic cardiomyopathy, then you get a checkup every 2 years. The list goes on and on. If you have a predisposition to breast cancer, then you either get more frequent and earlier mammographies, or you get prophylactic bilateral mastectomies and ovarectomies. And that extends to many other cancers where you might decide that the benefit of getting a piece of your stomach or your colon or whatever is at risk taken out in advance rather than waiting for you to develop a tumor. Sometimes people say, well, all I'm going to find out is I'm going to get generic advice to exercise and eat well. But the fact is, what's eating well for one person may be different for another person. So the Atkins diets is not generic advice, because if you have some kind of lipid problems, you're going to get ketosis. In fact, a lot of people get ketosis on a perfectly good diet for everybody else. And you could say exercise, well, some people should exercise and some people maybe shouldn't if you're at risk for some of these. So I think even what you might think of generic advice should be personalized. Maybe the way you get educated is something that we should consider, not to deny people education, but to customize it to opportunities that might exist. It'll be an interesting dialogue.
That article you flashed on the screen from the New York Times was provocative in its title, but underneath, within the article, was a lot of good information, I think, about the complexities that are being learned about what switches genomes off and on, and how complex the interactions are to get diseases to start or mitigate. You kind of didn't go into that very much. Could you indicate how your project interacts with those discoveries?
So the starting point for this article, which I mentioned, was I thought a fairly well balanced and thoughtful article. Who knows who picked the title for this? It's unfortunate some people just read the title. The basic point was letting the public know about these New England Journal of Medicine articles, which were responding to, in a way, the disappointing results that came out, or at least, I mean, it's something to be celebrated in that it is the biological answer, but the hypothesis that commonly occurring DNA variations, which were thought might explain a certain amount of the variation in the traits that we have. So common variants in DNA might explain common traits. That turned out that those common variants explained a very small amount of the common traits. And some of the people interviewed for this article and some of the ones that wrote the New England Journal of Medicine articles, they were saying that what it may be is we need to look at rarer, they were generally saying we need to look at rarer alleles, not just the most common ones, because the most common ones actually tend to be not very harmful. If they were harmful, they would be eliminated from the population, and that's what they were finding. So we need to go to rarer ones, and hence they need to be doing sequencing. So I think one of the take-homes was both this article and the story and the articles that it was based on, is that we need to be doing the sequencing. And that's what the Personal Genome Project is basically doing. It's doing sequencing rather than trying to look for the common DNA variants, which constitute 0.02% of your genome. That's what we were looking at, 0.02% of your genome. You really need to start thinking about looking at all of the genome. That's the major take-home, I think. Got 3 questions down here. Yeah, there you go. Yeah.
I was wondering if you could tell us about what you think the implications of what the whole personal genomics revolution are for the whole healthcare system in terms of the big 3 of access, cost, and quality. Thank you.
Access, cost, and quality, and maybe a few more words explaining that, what you mean by access, cost, and quality. You mean?
Basically, I want to measure by the effectiveness. When people look at the healthcare system, they look at, trying to look at access, the cost of care, and the quality of care, because they're all sort of related.
Right. So to some extent, there's been a reluctance to give quality personalization due to a kind of a fear of too much information. There was a fear that if you collected more information than you knew how to immediately act on, you would be held responsible for it legally at some later date. And I think this is one of those perverse social structures that really we all hoped would disappear. And I think what's happened is as the cost of the technology has dropped, and by technology I mean computer technology and now DNA sequencing technology, I think doctors and medical societies are getting over this fear of extra data, and that will open the door to collecting these very comprehensive data sets which will lower the cost, because now instead of each test being $2,000 for your inherited genome, maybe $100 for a microbial assay, $100 for the lipids in your. You can do these more comprehensive tests which will improve the quality, decrease the cost, and access is still going to be an issue. It's going to depend on exactly how we do the economics and all the socioeconomic problems. We typically get convoluted on this. But I think overall we're going to be aiming in a direction of greater access and lower costs. And there's something has to change in the healthcare system. This might be it. We don't know. We'll find out. It's an experiment.
There's a question here in the middle.
Yeah.
Yeah, I had a couple of questions anyway. I'm not, a geneticist or anything, but it seems like from gene expression or from the genetic information to the actual expression of the organism or whatever for disease or whatever, there are a lot of factors involved, like the epigenome and environmental insults, as you described. So in order to get a better picture of this epigenome or how it affects, genetic expression, having this really large sample size like you were talking about is important. But what about diversity in the sample? I mean, in terms of getting the genetic information from as wide a variety of genetic sources as possible, and also as far as the environmental effects and the environmental insults that may occur, getting a variation in that environment as well. Have you given thought to that? And then also, when you start collecting a better picture of this, system of genetic expression, going from the genetic information to, the organism expressing itself, how there might be an implication in terms of preserving the overall genetic health of the human genome, not the personal gene, but the human genome on the whole.
So certainly we want, we're trying to capture diversity in a variety of ways, working with different communities, say educational communities, disease-related communities, foundations, and so forth. Looking carefully through, when they go through the exam, they give us information about themselves before they actually get fully into the study, and we can go through that information and prioritize initially so that we're seeding nuclei in each of these communities rather than having too many focused on one particular disease or one particular, say, wealthy ethnic group or something like that. And by seeding those, then word of mouth and local press coverage or whatever can then take each of those nucleation points and they spread out from them. But a lot of this is an experiment, and we're looking for feedback on how to maximally benefit the maximum number of communities. So this will almost certainly change with time. Yeah. Question right there. Oh, here you go.
I have a few questions. Is that all right?
Pardon?
I have a few questions. Is that all right?
It's fine with me.
Okay. My first question is, what would you recommend for an undergrad interested in genomics?
Okay.
Should I ask all three at once?
Sure. I might be able to combine them.
Okay.
Yeah.
Also, what kind of careers are there in the field, and is it just research? That was one question together. And then my last question is if you think I should go to Northwestern or you.
Okay. Well, I'll start with the last one. I think Northwestern is a great place. You really should very seriously consider it. I think that biology and many fields are increasingly becoming computational. So whatever you do, add computational to it and study some of the social phenomena too, because this is not something that's just science. There's many opportunities. So in the Personal Genome Project, we have legal advisors, pro bono legal advisors, we have ethicists, policy. It's a very diverse project, and so there's opportunity. So think interdisciplinary is another thing to think. And I think that's all your questions. Yeah. I think there was one more back here.
One more here.
Yeah. Right, there you go.
Thank you, Dr. Teach. I apologize to the audience, I was late. I actually came from an integrative medicine conference that's being held downtown. And as much as you are revolutionizing how we think in medicine and in science, we're still not at the basic level capturing the data that we need in terms of lifestyle. For example, take something like how food practices have changed in India, given now that they're adopting more Western diet and the narrative that comes, as you mentioned, with interdisciplinary studies, the anthropological, the legal. So help me along. We get this personal genome, we get it, from cells, I'm a pathologist. And how are we going to capture that nutrition and lifestyle information? Because I wear that hat as well. I came through biochemistry and nutrition first, and then came to medicine. And we can't even get accuracy as far as, eating practices and consumption data and exposure data. And we have almost an epidemic of vitamin D, deficiency. So help me along how we're going to track this data, integrate it into the opportunities that come off of this SNP information, and then produce something that's robust that will give us practical, helpful, preventative, as the gentleman asked, solutions.
That's a really excellent question. And I think the answer is this project isn't going to answer every question. It's going to take the low-hanging fruit to start out with, but it's going to provide a framework which will collaborate with many other projects and enable some and hopefully draw a spotlight on the big problems like dietary diaries and maybe convenient personal monitoring of body fluids, and making these devices that you might have in your home so cheap that everybody can have them. They might actually become part of personalized medicine so that everybody expects to have these devices. Not just the diabetics having a glucose sensor, but everybody having these things. And once you get a mass market of people that are much more conscious about their biochemistry, it's got to be a positive for society, not just for the Personal Genome Project. So if we can provide a little spotlight on that, I know that scientists and epidemiologists are aware of this, but we need to get the public aware of this and get creative inventors, maybe even garage inventors, to start inventing the devices that you can carry around or have in your bathroom or that sort of thing. So I don't consider it a solved problem, and I don't even necessarily think that PGP is tasked to solve it. But we hope we create a kind of a meeting place, a watering hole where people can come, share data, share experiences, open source hardware, software, wetware, the whole thing. So great question. And I think we're going to end on that question.
Yeah, I think we'll end on this question. I would like to thank Dr. Church once again for an outstanding talk. And although he just mentioned this is not going to solve our problems, I think we all feel that we are at the beginning of a new era in biomedical sciences and how we will practice medicine in the not so distant future. Thank you very much.
Thank you. Yeah.